Hi, everyone. My name is Dina, and I am a recent graduate from the University of Windsor. And today I'll be talking about my project that I did for 2 summers under the Brain Tumor Foundation of Canada Studentship. And my project is Self-Immolative Nanocapsules, a new form of drug delivery for Glioblastoma multiforme. And for this project, I did it under the supervision of Dr. John Trent at the University of Windsor, along with my PhD mentor, Mana. So let's begin with a little bit of introduction. Glioblastoma multiforme, or GBM, is the most common type of malignant brain tumor. It's almost always lethal with a very low survival rate. The problems exist with surgical removal, such as it's really tricky to perform surgery on anything in the brain, basically, because if you remove a part of healthy tissue, that might cause some irreversible or permanent damage. So it's a very finicky process. It does respond to anti-cancer medications according to some studies, but there are problems associated with that, such as the tumor can take many forms due to genetic mutations, and they might not respond to the anti-cancer medication well in some cases compared to the other. So there are two main problems associated with targeting GBM or any brain-related diseases in general. So the first one is known as blood-brain barrier. This is a highly selective structure, or also known as a semi-permeable membrane that exists in our human brain. And the purpose of this membrane is that it only allows certain substances or chemicals to enter the brain, whereas other things are filtered and they stay outside of the brain. The second issue is that there's something called polymorphism. This means, so morph means like shape or form, poly means more than one. So obviously it means there's more than one mutations that might exist for GBM that causes it to be tricky to target because once again, they might respond to different medications differently based on how the mutation affects the cancer or the tumor. And these are in addition to standard treatment problems in pharmacology, such as premature drug release, meaning that a lot of the times the drug will release prematurely in human body before it reaches the target site, causing unwanted side effects such as cytotoxicity, meaning that the drug is now killing the healthy cells we want, the healthy cells that we don't want to kill, and causing some toxic effects to human body before it reaches and kills the cancer cells at the target site. So these are some common issues associated with targeting tumors or brain tumors in particular. And this is in addition to problems associated with some traditional drug delivery methods. So in this study or research, my group's focus was to design a new nanocarrier drug delivery system that doesn't exist currently in the world. And there are other nanocarrier drug delivery systems that has been developed, such as liposomes or micelles. Now, these are small capsules. Liposome has a phospholipid bilayer. That's what we call it. So there's like 2 layers of lipids or fats that come together, and they will be hydrophilic on the outside, meaning they can dissolve in water, whereas the inside of the membrane, it's the fatty tails that stack together, and they're hydrophobic on the inside. So this is really common. The other one, micelle, is basically just there's only the hydrophobic head that hydrophilic head that dissolve in water, and then the tail is once again hydrophobic, so repels water. So they kind of like form little clusters or little clumps in water, and then they protect whatever medicine is on the inside because the water doesn't break down micelle. It doesn't go inside. It will stay on the outside of it. However, issues with these two drug delivery systems or similar systems that it breaks down easily. So in chemistry, there's a term called entropy. We say that it is entropically favored for something like a bigger molecule to break down multiple pieces of smaller molecule because that creates chaos or disorder in the system, and it increases the overall entropy of the system, meaning that if it breaks down, that's like favored by the law of thermodynamics. It's the same for pretty much every single nano drug delivery system that has been ever invented or studied. As you can see in the picture, they can like kind of budge off and then form little capsules. And this is not ideal when you try to administer a drug because the drug can get lost in the body before it reaches target site. And then here's just an example of what the components are for liposomes. Micelle will only just have a monolayer or single layer instead of liposome having a bilayer or 2 layers. So the theoretical framework of our design is that we want to design a drug-bearing nanocapsule that shows stability during transport inside and outside of human body. because what's the point of making a new drug delivery method if it breaks down either during transport from a manufacturer or a factory, or sorry, big pharma company to a hospital, or during transporting a human body in the middle of a procedure. So with that in mind, we kind of had this approach, like targeting different angles of this issue. So there is a concept in chemistry called polymer, poly meaning more, and then so these are repeating units of the same thing. And the same thing is called monomer, so small building blocks. Think of a train or even like a string of pearls, like this picture has shown here. The little beads are the monomers. They are the singular units that compose and make the long chain. So our ideal is that we want to use a polymer as the backbone of the nanocapsule. And so you will have, imagine like 10 or 15 strings of pearls next to each other. But these are just strings. They won't form a ball or hold anything inside. That's where the cross-linking comes into place. So now we're tying little knots in between every single individual string, so they become a web, and they're kind of indestructible in a way. Once you have that backbone, you will put it into water or some solvent mixture, and then they would basically fold onto each other or fold onto itself and collapse into a ball and trap drug molecules inside the ball. That will be called the nanocapsule. And that's not everything, actually. There's more to it. We are designing to attach surface peptides, or basically anything else that researchers might want, to the outside of this capsule. And this is for cellular recognition. So when the drug needs to pass through, for example, blood-brain barrier, having those peptide sequences, peptides are smaller building blocks of proteins. They are like a sequence of like repeating units that sometimes proteins in like the berries in human body will recognize and say, oh, this is a part of our own stuff. We can let it in. So it won't be filtered outside of the barrier. So those peptides can be attached to the nanocapsule for cell recognition purposes, like I said. And then the best part of this, or two best parts. The first one is the use of novel self-immolative polymer backbone that degrades on command at tumor site. Because, well, if the drug is too stable and it doesn't release, it's also pointless. We can't have a drug that doesn't do its job. So the idea is that you want to build something stable enough during transport, but unstable enough at the target site. Once you apply a certain stimuli, it triggers the breakdown of this backbone. And I will explain more in details how that works in my future slides. And then the last thing is it has great biocompatibility, because the backbones that we choose, the monomer or the singular units, the little beads that we are using, are actually amino acids, which are building blocks of peptides and proteins. So they will be harmless to human body because those are things that you naturally find in your body. Okay, let's talk about the methodology of this work. Here are two diagrams I'll have to go through to explain things better. The first one is the mechanism of breakdown. So the entire idea of self-immolative polymer, which is a relatively recent field in polymer chemistry, we have repeating monomeric units attached by something called the linker to a triggerable functionality, or also known as the end cap. And then you will apply a stimulus. This can be many different things: heat, so temperature, acid or base, variations in pH. It can be enzyme triggered, or even light, because some molecules are destabilized by the presence of visible light. Once the stimuli is applied, the polymer will break down. The end cap will be cleaved off, and that will destabilize the entire structure, causing it to break down in a chain reaction fashion. So those little beads will just come off one by one because the linker will just cleave itself automatically. And the spontaneous depolymerization will give us small molecules and release whatever is inside the capsule, because now the entire backbone kind of just went through a detonation process and there's nothing left there to keep the drugs inside. So with this in mind, we still would like to use cross-linking because it makes that web-like structure, so they're not just individual like string of beads floating around. And this whole thing will be stable unless triggered. So researchers or medical professionals, if and when this is used in procedures, they will just control and apply the stimuli only when the drug reaches the tumor site to achieve controlled release at target site. And here is a structure of what the monomer will look like. Due to confidentiality agreements, and this work is still in progress. We are still in the progress of either patenting or publishing this work. Hopefully you will see it in publications down the road in future years. But so I can't really disclose the entire structure, but I can try to make it as close as possible. This is what the monomeric units will look like. So we have the amino acid backbone. This is a peptide. And then we have the end cap. So not every single monomeric unit will have an end cap. It will only be on the terminal ones because you only need one cap for a string of polymers. We will have a variable functional domain. Basically, this is anything that we want to attach to the chain or the capsule. So like I mentioned, could be peptide sequences for cellular recognition to get the drug bearing molecule through the blood-brain barrier, or anything else. If researchers want to vary that part and put even like drug molecules on it outside of the capsule, that's also possible because they're linked by chemical bonds. It will be stable. It won't really release it prematurely or not too much. This is the self-immolative linker. So those little black lines in between the red beads, these are the things that once the end cap is removed or cleaved off, the self-immolative linker kind of destroys itself to release the monomeric units in a chain reaction fashion. And the activation group is just something that we temporarily place to build the polymer. So this can be different chemical functionalities or functional groups that once you build the string of beads, they are gone because they were only there to make sure they connect. All right, we also thought of implementation of click chemistry, which is actually a really hot area of research topic as of late because this is the work that won the most recent Nobel Prize in chemistry. It's the use of click chemistry and bioorthogonal chemistry. What it means is that it's supposed to make chemistry very easy. Things are supposed to click together like how you're building Legos. They are not really affected by external circumstances. They are easy to perform, replicable, precision in like the work, and also bioorthogonal means that, because orthogonal means perpendicular, so they can also work in biological systems without really interfering the biological systems that it's placed in. All of these are very desired traits in terms of medicinal chemistry and pharmaceutical research. And here's just an overview of what the famous click reaction looks like. So azide, alkyne, copper catalyzed click reaction to form triazole. These are, high yielding reactions with no need of purification, simple to perform. And this is the textbook example, the QAC reaction. So we want to use the click reaction or click chemistry to perform the linking of those peptide sequences to the exterior layer of the nanocapsule. So we can just prepare an azide and then prepare an alkyne. So the capsule can either have the alkyne half or the azide half, and then the other part of this equation will be the peptide sequence, and you click them together and then they just stay like that. It's very easy and it's a great chemical reaction to be performed. So let's talk a little bit about precision in drug release. Now, the great thing about end caps or end caps for self-immolative polymers is that it's only responsive to specific stimuli and not anything else. So the common ones, like I said, are temperature, which is heat, and then you can change the acid or base level in human body. However, that might be a little finicky. That's why we're not going that route, because there are parts in human body that is more acidic than other body parts, like the stomach, and there are parts that's more basic than other body parts, like the intestine. So it's really relatively hard to control the pH level in human body and make it like stable to make sure the drug releases at that spot, because there's other spots with similar pH levels possibly. Enzyme, that's another trickier one. That means you'll have to have specific enzymes or proteins to cleave the end cap. But once again, in human body, that's something you don't really have control over, because there might be many enzymes floating around the bloodstream, or I don't know, the lymphatic system, that's hard to just precisely target. So our choice of stimuli is either one, heating or magnetic heating, and the second one, light. We first wanted to go with magnetic heating. This is a method to generate heat in a relatively small area using vibration, and it's already been used in some procedures, so it's safe to use. I believe it can generate temperatures up to 60 degrees, if I'm not mistaken, degrees Celsius. So this will cause decomposition of the self-immolative polymer on site once the nanocapsule is at the tumor site. So it's very not really harmful to human body and stuff like that, which is ideal. It also prevents premature drug release because other parts of human body won't necessarily have that high of a temperature to cause the capsule to break down. So it's very controllable. And when it comes to light, this one might be trickier, but we can test this. It's in pilot like studies where zebrafish have transparent skin. So if you put something in a zebrafish and then keep it in the dark and then you shine a light on it, you can actually do studies to monitor that the drug release rate in real time inside zebrafish because of the light now is that is now present. But that's so the magnetic heating one, the heat triggerable end cap, we already have made it. The light sensitive one, it's still in the making at Trend Lab. So when it comes to result and conclusion, once again, I couldn't really disclose the structures, but I can try to verbally describe it as close as possible. We have synthesized 2 different types of polymer backbones successfully. And these are the variations is at the activation group. So by attaching one functional group versus the other, we can see how fast the monomers polymerize and how good they decompose once the trigger is applied. It's really, this is called kinetic studies. It's really useful in chemistry because sometimes maybe you want the drug to release faster, while other times you want it to release slower. So by varying the polymer backbone, actually 2 different types might be a mistake. I think there are 6 different types that we made. We use 3 different amino acids and then 2 functional group each for activating. So that's 2 times 3 equals 6. We made all 6 and the procedure is easy to follow, can be done. It's relatively like reproducible chemistry. And then the first summer, I synthesized thermally sensitive self-immolative polymer with my PhD mentor Mana, and then she performed the degradation studies, meaning you then now can like see how long the polymer is, how many repeating monomeric units there is, and how they respond to the stimulus that makes them degrade. And then the second summer of my studentship, we were trying to pursue the second step of the project, which is synthesizing the light-sensitive end cap. And the chemistry there was relatively interesting because I had to perform a lot of reactions in the dark, going to lab close to midnight when no one else is around, turn off all the lights, sometimes night vision goggles, and then just do chemistry in the dark. It was quite a unique experience. And that light-sensitive end cap did face some difficulties because it is kind of unstable. You have to really control the light that goes in to the reactions, and sometimes it can decompose if you place it like longer than intended. So it kind of has to be followed up quickly. So this is still giving us some difficulties, but we will overcome it. I'm faithful to that. And the next step will be self-assemble the polymer backbone, meaning now we have the strands of beads. Once again, going back to that analogy, we want to cross-link them, make them like tie them up, little knots in between the chains, so they are a strong web, and then put them in solvent systems so they collapse back on each other to form the capsule. Also, in the same time, there will be drug molecules floating in the solution so they can encapsulate the drug inside. And then with that capsule obtained, we will then hopefully perform in vitro and in vivo studies to see the degradation rate, see how it's released, see how it affects the zebrafish in real time, etc. And I'm really excited to see where this project goes in the future. So at the end, I would just like to say thank you to everyone that has supported me in this research project, and that is the team that made me into the chemist that I am today, the Trend team at UWindsor, and then thank you to Brain Tumor Foundation of Canada for giving me this opportunity and the studentship so I can pursue such a cool study that hopefully we will see the application in real life years down the road. Thank you also for listening, and if you have any questions, please feel free to reach out to Sue at the Brain Tumor Foundation of Canada and she can forward your questions to me. Thank you and have a good day.